different is this morning? Indeed, very wonderful, and the waves around tell us the beautiful tales. The waves around tell us the with is truly unforgettable. Myself, here Inuka, from 2nd BSc Microbiology, shall be your host today. I welcome you all this wonderful gathering. It's my great pleasure and the first and foremost duty to warm welcome acknowledge the presence of her honorable welcome speech.
Good morning to one and all. I am Sandhya from 2nd BSc Microbiology. Sometimes challenge and struggle are exactly what we need in our lives. May you welcome every effort, every struggle, and every challenge, and may you open your wings and fly. I welcome our Honorable Chairman, Sir, Dr. M. Karnanidhi, Sir, Vivekananda Educational Institution. I welcome you, Sir. Today, I am very happy to tell you that we are celebrating the webinar on soil and plant microbiomes for sustainable agriculture. Next, I feel extremely honored to introduce the chief guest, Dr. G. Kabildev Sir, Assistant Professor, Department of Microbial Biotechnology, Bharatiyar University, Coimbatore. I welcome you, sir. And we are proud to welcome our principal, Dr. P.T. Suresh sir, and academic director and IQAC, Dr. B. Kumaravel sir. I welcome you, sir. I am very glad to welcome our supporting pillar, Dr. T. Shankar sir, Dean of Biosciences and IQAC, and Dr. L. Asokumar sir, Head of the Department of Microbiology and Department of Nutrition and Dietetics. I welcome you, sir. And special and glad welcome to this webinar organizes our Miss M. Yuvrani ma'am, Assistant Professor, Department of Microbiology. Welcome you, ma'am, who always encouraging and motivating us. Be a wonderful role model because you will be the window through which many children will see their future. I welcome our beautiful faculty members who always guiding us, motivating us. I welcome you, ma'am. I welcome you, sir. Last but not least, I welcome my dear friends for this wonderful session. Once again, I welcome you all. Thank you.
Thank you, Sandhya. Is on her side today. The Sanan as pleased to have offered you all warm welcome. I'm profusely overjoyed to take the opportunity to introduce our chief guest, Professor, the Department of Microbial Biotechnology, Bharatiyar University, Coimbatore. Tell me and I forgot, teach me and I may remember, involve me I learn. Here I welcome her most respected head of our department, Dr. L. Ashokkumar sir, for the brief description of our guest of honor.
Thank you very much for thank you very much for your nice introduction. A warm good morning to all the gathering here. I welcome. First of all, I welcome my faculty, fraternity, and all of my students for today Department of Microbial Biotechnology, Bharatiyar University, Coimbatore. All the university, UGC, and DBT, and University Research Fellowship in Bharatidasan University, He passed the set exam in the year of the 2016. He got the more than 24 lakhs research fund from the various funding agencies like the UGC and RUSA, and he attended the more than the 12 national and international level seminar and training proceedings. And so, dear students, this is a very important day, and nowadays is a very important topic also. So, the lesson, the wonderful speech from the wonderful and experienced personality without any disturbance, and accurately the more knowledge about the sustainable agriculture and from this webinar. And thank you all of you. Thank you very much.
Thank you, sir. I feel so grateful to call her chief guest Dr. G. Kapildev sir to head over the session for the lecture.
Thank you. First of all, I good morning, one and all. I'm very glad to present on this topic, soil and plant microbiomes for sustainable agriculture. So before that, I thank for your nice introduction. And also before my presentation, I wish to thank the management authorities, Professor Dr. M. Karnanidhi sir, Chairman and Secretary, and Head of the Department, Dr. L. Ashok Kumar, and the Dean Bioscience and IPSC, and Principal Sir, Dr. B.T. Suresh Kumar, and the Director Academic and IPSC, Dr. V. Kumarvel Sir, and the Coordinator, Mrs. M. Yuvarani Madam, for your invitation to present the topic on soil and plant microbiomes for sustainable agriculture. Thank you. And I may warm welcome to all the participants for this webinar. Hello, ma'am. Am I audible? Participants?
Yes, sir. Yes, sir.
Yes, yes. Sorry, there is a little
It's okay, sir.
connectivity problem. So again, I repeat that I thank for your nice introduction, and I thank our secretary and chairman, Dr. Professor Fernandez sir, and the principal, dean of IPAC and academic, and the head of the department, and the coordinator, Mrs. Yuvarani ma'am, for this nice opportunity. So shall I start, ma'am?
Yeah, sir. You can start, sir.
Yes. Ma'am, is it my PPT?
Yes, sir.
So today my topic is soil and plant microbiome for sustainable. So the participants, I'm not going to take anything. And this webinar is mainly to think about the future problem, and the issues is the need of the hour, because we are facing a lot of problem, because we are not doing any sort of agriculture practice in a proper way. So in that aspect, how these microorganisms help for the sustainable agriculture. So that's why to make you to think, and you need to change, because you are the young generation. You have the power to change the society forever. Mainly we are dependent on this agriculture for our global food production. So there is a demand in that. So here we have to discuss how to rectify the problem, and how we are going to feed the global meet the demand of the global food production. So the problem is worldwide growth in population increases in the demand of global food production, and the environmental damage causing problem in agriculture yield or major concern to the world. So one side the population getting increased, and another side the land area for agriculture is getting decreased. So this is a major problem. So in that available space, how we are going to produce food from agriculture for the global demand. So far we did a very good what to say, that practice of traditional agriculture. It was very self-sufficient and everything. Now we are totally deviated from this traditional agriculture practices, and we are not bothered about the ecology and the ecological balance and the environmental issues. So due to the continuous advancement in the technology, science and technology, we are moving fast to in search of some other thing, but really we are not bothering about our traditional way of anything. It may be a culture or tradition of agriculture. So in that aspect, what are the lagging things, what we need to do, and what is the current situation about this agriculture, and how we can recover this. So these are the things we are going to discuss in this topic. So we know very well that microbe, soil and plant have a very good relationship. So most of you from microbiological background, so you know what that plant microbe soil interactions. So they are interdependent for everything. So microbes lives in all places. Here we are going to in the point of view of agriculture. So the soil having the lot of microorganisms and the plant, how they interact, what is their need, what for they are interacting, and what we are getting. So that thing we are going to discuss. So agriculture itself the world's most important technology because this is one of the very old technology. Still means our ancestor they practiced from several years back. So this technology nowadays get advancement and deviating from their traditional. Back
Sir, your voice is not audible, sir.
dear participant, sorry for the here we facing poor network connection problem. Am I audible?
Yes, sir.
Okay. So we are discussing about microbiome, or everywhere symbiotically helps sustain life in all its forms. So it is present in all almost all part of the plants. So this is a plant microbiome association. Plant microbiome represents the complex microbial communities associated directly or indirectly with the plant. So we can call it as a endophyte, epiphyte, and the rhizosporic microbiome. So endophytes are present organisms present inside the plant organs, and the epiphyte present in the surface of the plant, and the rhizosphere present inside the rhizosphere area. Rhizosphere zone means that is a root area of the plant. So these microbiomes are interact with each other for their growth and development. Plant microbiome involves pathogenic as well as non-pathogenic microbiomes. So non-pathogenic includes neutral as well as symbiotic members. So they are all root, stem, leaves, seeds, pollen, like that. So then rhizosphere microbiome present in the root and is present in all soil. So there is a difference in soil microbiome and rhizosphere. Rhizosphere microbiome present near or nearby the root area, that is root zone. But in soil microbiome, that is present in all part of the soil, all region of the soil. So how does we assess the microbiomes? So microbiomes present in everywhere. What is the need and how to assess? So normally we used to collect the sample from the any part of the plant or from the soil. So need to extract the DNA to identify, that is, I'm talking about the molecular identification. So we can identify through various techniques. So I'm talking about the molecular identification. We can extract the DNA using any other protocols, and we can amplify using PCR, that is 16S RNA for bacteria and 18S RNA for fungi, ITS, like that. Markers we can barcode markers we can use to identify these microorganisms. So there are a lot of advancement in the technology. We can amplify, we can sequence, and we can identify who is there, means what are the organisms present in which part of the plant. At the same time, metagenome sequencing, that this technology used to identify the uncultured, means unculturable organisms. That is, and then we can also identify their functions, what they can do inside the plant or in the soil, like that. And community RNA, protein metabolic, we can also isolate that RNA, protein and metabolic extraction, and we can study that metatranscriptome sequencing, metaproteome analysis, and metabolome analysis. So this community activity and what they are doing, everything we can easily assess using the this nowadays we have a very good high throughput technology. We can assess easily. Here application of plant associated microbes. So there are a lot of applications that association with the plant and microbes, that is biotransformation, biodegradation, phytoremediation, seed production, seed predation, plant growth promotion, stress tolerance, biocatalysis, biofuel production, biocontrol, agricultural importance, source of novel natural products, biosynthesis, and many more. So these are the lot of associated microbes, plant associated microbes. They will do this type of job in the plant system. So this plant microbe interactions. So already we discussed about the epiphytic and this endophytic everything. So they mainly they protect the plant from the predators and even from the pest or from the disease, means pathogenic organisms. So that is induced systemic resistance and systemic acquired resistance. So whenever any pathogen will affect near to the plant or trying to infect the plant, so these epiphytes or endophytes interact with them and encounter the pathogens by secreting any other second metabolites or any other compounds. Okay, so this what way they both are benefit means we can say that carbon root exudate attract heterophytic microbes. So some exudates of the plant through the root will attract the microorganisms that will be used as nutrients for the growth and development of the microorganism. From the microorganism side, plant also get a lot of various process already we discussed in the applications. So it can mainly it will break the heavy matter into the simpler utilizable form, that is ammonification, nitrification, like that. The main thing is degradation process. So and it will readily make it available to the plant. And both are going on mainly to protect the plant, and they both are benefited without any harmful without any harm. So this
Sir, voice is not audible, sir. Girls எல்லாரும் வெயிட் பண்ணுங்க, நெட்வொர்க் இஷ்யூஸ் இருக்கிறதுனால கொஞ்சம் லேட் ஆகுது. சரியா?
sorry again face a severe network problem here. Sorry for the interruption. Hello ma'am.
Sir,
Network problem. இப்போ screen share ஆயிருக்கா மேடம்?
sir,
Screen
இல்ல
is
இல்ல sir.
visible or not ma'am?
இல்ல sir. It's not visible, sir.
Actually host disabled participant screen sharing இருக்கு மேடம். நீங்க ஏதாவது disable பண்ணிருக்கீங்களா?
No sir, I will check, sir.
ஷேர் பண்ண ஓகே.
Sir, நாங்க எதுவும் பண்ணல சார் இங்க.
Yes ma'am. Actually host disabled participant screen sharing காட்டுது. That's why இப்போ screen display ஆகுதா மேம் PPT?
இல்ல சார், டிஸ்ப்ளே ஆகல சார்.
Again the same post disabled participant screening. I'm not able to share my PPT.
Sir, just a minute, sir. கீழே co-hostல தான் இருக்காரு சார். இல்ல மைக் ஆன் பண்ணி சொல்லுங்க சார். இப்போ ஸ்கிரீன் ஷேர் பண்ணுங்க சார்.
Yes, ma'am. Now it is visible.
Yes, sir.
PPT there. Okay, okay. Thank you. So we were discussing about this plant microbe interactions. So they both are mutually benefited, the product of the plant and the product of the microorganisms. So they both sharing each other, and they are living a very good means without disturbing anything. They are benefited. So what we are trying to say that we need to support a minimum support to the plant, because they have a well-defined ecological balance between this plant and microbes. So there is a problem in this nowadays. We are continuous using of any chemical based fertilizers or chemical based pesticides. So pollution, soil pollution, and air pollution. So together, mainly the pollution problem, together combinedly, they affect this ecological balance. So that the in the plant as well as in the soil day by day getting decreased, and that's about to extinct. So this is now the problem in the modern agriculture. So the modern agriculture, we are using lots and lots of fertilizers, chemicals for benefit of plant, but indirectly it will not protect the plant. Not only kill the pathogens, it also kills all the essential or beneficial organism present in the soil as well as the plant. So here the ecological cycle means imbalance arise. So to avoid this, we need to avoid the minimal use of, not even a minimal, we should not use any chemically synthetically prepared chemicals, that is a pesticide or fertilizer, everything. So only the use of natural and organic manures that is advisable to the plants. So we can compare with the crop plant and the wild plants. So the wild plants, we are not giving any support to the plant system. So even though they are producing, they are growing very good healthily, and they bearing fruits, everything in the wild condition, that is in the natural ecosystem. But in the crop plants, we are facing a lot of problems, that pest, disease, abiotic stresses. That is because of the ecological imbalance. There is the absence of soil microorganism, that is beneficial organism present in the soil as well as in the plant. So both get almost they are spoiled or degraded in their ecosystem. So this is the current situation. So we need to enrich or develop this microorganism in the ecosystem. So how it is possible, that we will discuss. So normally this is the interaction, protection against pathogens, immune response, tolerance to abiotic stress, physiology and metabolism. So almost all the protection or growth and development, everything they are doing their own. So no need of the man interruption for any crop production. The thing is, so what we are doing, we are unnecessarily using unwanted chemicals and pesticides in no recommended dosage, and we are the farmers, they unaware of using this, how to handle these chemicals and the fertilizer, pesticide, they are just simply throwing and using this. Because of this, the total ecosystem got damaged. So role of plant to play important role in soil improvement, plant growth promotion, stress resistant. They are bestowed with the distinguished feature of atmospheric nitrogen fixation, bioactive metabolites, phytohormone production, plant disease suppression, nutrient cycle enhancement, and many more. So, so as I told already, so the soil improvement also, we no need to do anything, no need to give any external fertilizers or anything. Only they themselves improve the soil by degrading all the heavier particles into simpler, and they maintain the soil pH, everything properly. And also it will develop this plant growth promotion, and it protect the plant in all type of stress resistance, either biotic stress or abiotic stress. So, and also they fix the atmospheric nitrogen, bioactive metabolites, phytohormone production. Sometimes it will help in production of phytohormones for the growth and development of the crop plants. Plant disease suppression, it will involve in the defense mechanism, and the nutrient cycle enhancement and meaningful cycling of the nutrients. So everything will be done by this microorganism present in the soil and also in the plant. Advantages. The microbial inoculants offer several advantages as they are more safe, have reduced mental cost, have less negative impact on human health, are active in small quantities, have many more positive applications. So why we are saying that more safe in the sense, so they are not a harmful organism, they are beneficial to the, and also eco-friendly. All these organisms are eco-friendly, there is no any harmful chemicals, so they are more safe. And then the cost of production is also very reduced, and there is a very less negative impact on human health. It won't have any negative impact on the human health as compared to other chemical fertilizers or chemical pesticides. We need to give a very little quantity of inputs, and which having the most positive applications in the soil. These products can possibly be used as biofertilizers. Nowadays, biofertilizers available in almost all in the market in the local dealer. So we can use these biofertilizers based on microorganisms or consortium of microorganisms and biocontrol agents. So instead of pesticides, for any chemicals for controlling the disease, we have very good biocontrol agents, plant thinners, phytostimulators, and biopesticides. So means it is a well-established fact that the plant cannot survive in the absence of microbial association. So that's why the tobac in the plant means in the agriculture, almost all the farmers are facing the same problem, because the problem is there is a lack of microorganism, there is a lack of association in the soil and the plants. So that's why the plant are struggling to survive in the absence of microbial associations. Therefore, deep understanding of the plant microbiome as a whole is essential in order to explore the same for better and sustainable agriculture. So we should study more about this interaction. Already there are a lot of studies are going on plant microbe interactions. So we need to study more and more to understand each and microorganism, how they are interact, and in what way they are benefit to the plant and the soil. That we need to explore more, and then only we can go for we can do this better sustainable agriculture. So the sustainable agriculture is nothing but already we are practicing in the olden days. This is traditional way of agriculture, method of agriculture. So nowadays we switch over to this modern agriculture. Here we are facing so many problems. So microbiomes of plant and soil. So as I told you, this plant and soil is a hotspot. So the plant and soil microbes play important role in plant growth promotion, soil fertility for sustainable agriculture, and also this plant growth promoting organisms. So they are also doing promising tool for important and promising tool for sustainable agriculture. So what will do that it will interact with the plant that is in the endophytes and the soil microbes. They promote plant growth, protect the plant from deleterious pathogens that the root exudates, inhibit or kill the soil pathogen through the production of phytotoxin. So whenever the pathogen approach to enter into the plant will produce some plant or the plant associated microbes. They release some secondary metabolites or root exudate, which will kill or arrest these soil pathogens, and the rhizosphere complex and diverse interacting ecosystem. So the plant carbon photosynthesis are secreted as food for the soil microbes. So the exudates, the nutrients released from the plant roots are used as a food and feed for these microorganisms. Root secretions also benefit plants directly by mobilizing plant nutrients. So these are the most of the major interaction between the plant and soils. So there are a lot of microorganisms present in the soil. These are some of the notable species, genera of microorganisms. That is Archimobacter, Arthobacter, Aspergillus, Azospirillum, Acetobacter, Bacillus, Bacterium, Penicillium, Pseudomonas, Rhizobium, Streptomyces, Mycorrhizae, etc. These are belongs to different genera. These PGP plant growth microbes can be used as a biofertilizer, bioplants to replace the chemical fertilizers that we can call it as sustainable agriculture. So there are a lot of alternate source to replace the chemical fertilizers. So these are based on this biofertilizer using this genera or the consortium of these microorganisms. Microbes plays vital role in the terrestrial ecosystem and the soil health. So the both thing that plant growth, plant health and the soil health can be maintained by their own because of this interaction between the microorganisms, either bacteria or fungi. So they are both are maintaining all sorts of health in the soil and plant. And the role of PGP microbiome, again that's endophytic bacteria. So it will allow the entry of bacteria through the root hairs and the rhizosphere bacteria. So bacteria residing in all, in the endophytes present in almost all part of the plant and doing their helping in their function, growth and development, and phytocarbon production, nitrogen fixation, ammonia production, ceterophore production, phosphate solubilization, and hydrogen cyanide production. So these all the activity are done by this organism present in the soil and plants. So again the same the type of PGBR mechanism. So either plant growth promotes direct or indirectly, they will help in the plant growth promotion. Mainly that antibiotic in the direct, ceterophore chelates ions, phytocarbon production, asymbiotic nitrogen fixation, phosphorus solubilization, indole IAA production, ethylene production, cytokinin, and that is phytocarbon production. And the indirect plant growth promotion, it will produce antibiotic and lytic enzyme, ammonia production, induced systemic resistance, HCN production, competition, and exopolysaccharides. So this is the mechanism of growth promotion by phosphate solubilizing bacteria. So how they promote the growth by solubilizing the phosphate present in the. So the main thing, the insoluble phosphate will be converted and it will make into soluble phosphate, which is readily available for the plant. That will be uptake by the plant, and they will do all these metabolic processes. So again, the phosphate mobilizing microorganism. So there are several phosphate mobilizing organisms are there. So there they will do their mineralization, translocation, solubilization, like that. Using their enzymes, they can degrade and they can convert into any other form. The main thing is they will make everything readily available to the plant. The three main process of phosphate mobilization and mechanism used by phosphate mobilization microorganisms that orange arrow represent potential phosphate mobilization by organism associated with fungal hyphae. That is so these microbes reduce effect of drought, flood, and so again this picture shows how they interact with all the abiotic stress. So these microbes enhance root growth via phytohormone production, more and deeper roots. Microbes minimizes the inhibitory effect of ethylene on plant growth, produce ACC deaminase, and microbes from biofilm that reduce ion movement into the plant. So in that way, they are very much helpful in to reduce the effect of these abiotic stresses like drought, flooding, and salinity. So here the biotic influences and the abiotic influences. Association in the rhizosphere between plant roots, microbes, and root exudates under biotic and abiotic influences. So during any stress, either biotic or abiotic, the plant and this microorganism, they together, they will encounter this problem and they produce certain chemicals or exudates or any other metabolites to overcome this problem. So mainly their interaction is chemotaxis, recognition, adherence, colonization, and growth, and biocontrol. So it will suppress the pathogenic effect, resistant to biotic and abiotic stress, reduced invasion of toxic matter. So increases solubilization and uptake of important macronutrients, improves fertility of soil, increase in yield, growth, and productivity. So according to the situations, the plant and the microbes, they will interact, they will communicate, and to produce this type of, they will involve in this type of activities. So how they will communicate? Mainly through the chemical signals released by the plant or released by the microorganisms. So where the interaction between plant and microbes living on the root surface or near the roots with the help of soluble molecules. So it may be in the interaction, if it is away from the root, it will interact with the volatile molecules, and nearby means it will interact with the soluble molecules, and the interactions with microorganisms living inside the roots. So this interaction takes place in soil, which is composed of rock, air pores, and organic matters. So wherever it may be, that all the organisms near to the plant root or inside the root or away from the root, everything will be communicated through various mechanisms, mainly chemical signals. So plant and microbes help each other to get food. So here one example, that is whenever the plant need nitrogen or any other means sugar and nitrogen. So they will release some chemical signals. So that is isoflavones. So that nod factor that the root hairs, that rhizobium will interact or enter into the root hair, and it will form a nodule. From the nodule, they will fix atmospheric nitrogen or any other sugars, everything. So they will give one type of interactions present in these legumes, mainly in the legumes, the rhizobium and the legume plant, they interact for their nitrogen source or sugar source. Main plant growth promoting groups used in commercial bioinoculation and various mechanisms each employed to promote plant growth. So nowadays we have a lot of use plant promoting organisms either in a single species or in the consortium form. Their role is stimulant, produce hormones for growth, example auxins, and nutrition, increase rooting area by supplying nutrients, nitrogen fixation, and phosphate mobilization. Then bioprotection, that is protect against from plant disease, and bioremediation, sequestering harmful substances, that is detoxification. So these all will be done by these organisms mentioned here. That is a lot of genera and species names are mentioned here. So they will do, and mainly this mycorrhizae, that ectomycorrhizae and the their role also very immense in this that the associated roots. Sometimes in the drought hit area, this mycorrhizae or the fungi, they will means they will help to take water from some other water resource. They help the root area to get water from the nearby water source. Like that, they have very good organized functions, means interactions with plants. So microbial consortium and its benefit. A group of different species of microorganisms that act together as community. So we can call it as different species of organisms act together as a community. This microbial consortium fertilizers contributing significantly to plant adaptation to various biotic stresses in extreme habitats, that is drought, salinity, heavy metal stress, pH stress, heat stress, pollutants, nutrient deficiency, and hydrocarbon stress. So these microbial consortium are used to avoid these type of stresses. So here are some examples that relationship, that is beneficiaries, botanical beneficiaries, that is bacteria and fungi that living in on plant can help their host in several ways. So here are some few examples that in plant legumes and other plant is rhizobia. Provide the plant with the extra nitrogen, which boost photosynthesis and help the plant producer bitter nitrogen containing alkaloids, because this bitter taste nitrogen containing alkaloids. So in that way, that defend against the predators, the plant, those who are animals that are eating plant, means they will not feed because they are producing some bitter nitrogen containing alkaloids. So this one type of interactions. And in another plant, maize, that is same the plant that is danction, that is Codonophyes pilosula is a medicinal herb known as a poor man's ginseng. So this Bacillus subtilis, this organism improve plant size, chlorophyll content, and photosynthesis. Another one is in a plant black cottonwood, Populus trichocarpa, that stachybotrys, reduce the severity of leaf rust caused by the fungus melamspora. Melody help the plant. So these organisms in the freezing also. So there are several. This is a few examples that I mentioned here. So what type of organism and what type of crop and what is their effect on that. Is really one plant we can't say the single group of the composition each and every part of the organism means in the plant. So these leaves, fruit, flowers, stem, root, everything. So here the chart showing that the composition of the organism varies in different part of the different organs. Okay, so for example, in bottom leaves, this rhizobiaceae, that is rhizobium, is almost somewhat 45%. So
Sir, again your voice is not audible, sir. கேர்ள்ஸ் யாரும் லெஃப்ட் ஆக வேண்டாம், லிங்க்லயே இருங்க.
Hello, ma'am. Visible.
Your screen is not visible, sir.
Hello, ma'am.
Sir, Sir, now your slide is visible, sir.
Okay, ma'am. So, sorry for the interruption. That microbiome can protect plants against pathogens or pests. Plants are subject to infection by diverse microbial pathogens as well as regularly by insect and nematode pests. So, another this insect microbe interactions with the plants, that is insect microbe and plants. So in one somewhat interesting thing that bacteria associated with the Colorado potato beetle manipulate the plant defense to facilitate beetle feeding. So this they have such kind of interactions, so that bacteria that symbiotic bacteria present in the this beetle will they have the ability to change the defense. They manipulate the defense mechanism of the plant, so in that way it will help that beetle for feeding. So this is one type of interesting work published in PNAS. And the sustainable agriculture produces abundant food without depleting the earth's resource or polluting the environment. So the term sustainable agriculture, everything. So we are the practice of doing the agriculture steadily without damaging or making harm to the environment. That is sustainable agriculture. So in that way, this microorganism present in the soil as well as the plant, they can do all sort of beneficial things to the growth and development of the plant and the soil health. So these are the some of the enable agriculture, irrigation, people and society, waste recovery and management. It will increase the economy, crops and animal health, earth and its environment health. So this a lot there. This is the different interactions using this sustainable agriculture. Benefits of this sustainable agriculture. So benefits of sustainable agriculture, again, that it will preserve the biodiversity, avoids deforestation, protect health and safety of community, protects economic viability of farming, and ensures the soil conservation, maintains water quality, suits local conditions and capacity. So by doing this sustainable agriculture, there are a lot of benefits. So that all the benefits will not harm any other impact into the environment. So we can skip this slide, and coming to this insects, approaches, challenges, and applications. Here we need to exploit these plant microbes or soil microbes for our sustainable agriculture, because now, as I told already, the continuous usage of harmful chemicals and pesticides, fertilizers, what are the chemical used to inputs given to the soil and plants in the modern agriculture, they have the negative impact on these microbiomes. So for that, we need to wild and domesticated microbes, plants feedback, host mediated microbiome agriculture practices and approaches, integration of multiomics, in situ detection of metabolites, network analysis and microculture. So we can recover and we can do some modifications work, or we can conserve these organisms by identifying their functions. And the challenges: temporal and spatial dynamics of microbial communities, multitropic interaction in the plant microbiome, environment influence on plants and microorganisms, and new plant breeding strategies improve crop yield and quality. So we are in the situation to exploit these organisms present by either in the wild or in the domesticated. We need to cultivate more, and we can use in the sustainable agriculture in the form of biofertilizers or biostimulant or consortium of microorganisms. Future strategies for plant breeding should take plant microbial symbionts as a lifelong bodyguards into the consideration. These proposed in this interaction approaches will provide solution for exploring and exploiting the plant microbial interactions for improving their sustainability and the productivity of global agriculture. So this future perspective. So then each and every year they have that populations also increases, the demand for the agricultural production also get increases. So now we are in the 2015 to 25, sorry, 2022. The last thing we can see that recarbonization of the biosphere, nutrient sensitive agriculture, soilless agriculture, phytobiome management, urban sky farming, space farming. So due to the lack of agriculture land and need to revive and update all this sustainable agriculture in different ways. So nutrition sensitive agriculture, we can give the such type some type of microorganism specifically for some particular functions. For example, phosphate solubilizing. Based on the need of the plant and soil, we can give this microorganism. And soilless agriculture, that is hydroponic system of agriculture are now in boom. So they are we here the only water is enough, water and nutrients is enough. There is no need of soil. Like that, they are urban sky farming, space farming. So there are a lot of advancement in the farming system using this sustainable agriculture. And another future thing is smart microbiomes that detect and treat disease or destroy pests. So this is one of the imagination, but this is possible because so some type of organism they can detect and treat the disease, that is antagonistic activity. So that can be used in the smart agriculture. That is so coming there global populations of 9 billions by 2050. And there is an urgent need to explore and understand the hyper diversity as well as functional potential of these microbial communities, not just for the sake of sustaining ecosystem services, but to maintain the beneficial use of biodiversity to mankind. So for sustainable development of the human world, the sustainable agriculture is the need of the hour. So with this, I conclude. Like, sorry for the participants. There is a problem in internet connection, so there will be 2-3 times interruptions in that. So sorry for that. And if you have any queries related to this, the thing is, the message I want to give to the youngsters is, agriculture is our age-old practice. So there is no gender, means this one not only for men or women. So both they practiced this agriculture in olden days. So now in this era also, in this time also, we can try to do this agriculture in there. How much land or how much space we have, we can do, and we can do it ourselves using this microorganism. Means that is a traditional way of agriculture. So try to please avoid chemical fertilizers, chemical pesticides to protect our environment and ecological balance. Thank you, one and all.
Thank you, sir. Dear students, now you can ask questions.
Participants, if you have any queries, you can ask. Feel free to ask. Participants, are you there?
ஓகே சார், நம்ம கன்க்ளூட் பண்ணிக்கலாம் சார்.
Okay, ma'am. Okay, ma'am. Participants, if you have any further queries or anything, you can contact me. Here in that my slide, you can see that my mail ID and phone numbers are there, so you can ask anything at any time. Okay, and thank you, ma'am. And again, I thank all the management and principal, head of the department, and the directors, dean, and the coordinator of this webinar, Mrs. Yorani, for this opportunity. Thank you, one and all.
Thank you, sir. Sir, just a minute, sir. Girls.
Thank you, sir. We are looking forward to having your speech again. Now it's time for vote of thanks. Hereby welcoming B. Jayashree, second BSc Microbiology, to thank with her pleasant final concluding words.
I deem it a great honor and privilege to propose the vote of thanks on this memorable occasion. Thanking you for giving these opportunities. First of all, I would like to thank our chairman, Sir Dr. M. Karnanidhi, Sir, Vivekananda Educational Institute, for giving permission to conduct this program. Even he is absent, his blessing is always with us. Next, I would like to thank our special chief guest, Dr. G. Kapildev, Sir, Assistant Professor, Department of Microbial Biotechnology, Bardiyar University, who, despite his busy schedule, has found time to grace this occasion. Thank you, sir. Today we had the opportunity to hear your thoughts. This is definitely encouraging us in our future events. And I also express my heartfelt thanks to Dr. V. Kumaravel, sir, Academic Director, for his valuable contribution. Thank you, sir. And I thank our well-wisher, our principal, Dr. B.T. Suresh Kumar, sir, Vivekananda College of Arts and Science for Women, Autonomous, for support, vision, and commitments. Thank you, sir. And my heartfelt thanks to the Department of Bioscience, Dr. T. Shankar, sir, and our head of department, Dr. L. Ashok Kumar, sir, who have worked hard to ensure that this occasion become a memorable success. Both of you, thank you, sir. We are grateful to our faculty member for his words of encouragement. His able guidance has always encouraged us. I thank Ms. M. Yuvrani ma'am, Assistant Professor, Department of Microbiology, and I thank him for his kindness. Thank you, sir. Thank you, ma'am. Last but not least, I would like to thank my friends for your cooperation in making this function a resounding success. Once again, I thank all the members involved in organizing an event, including the guests who have a part of the event. Thank you all for your attention. Thank you.
Okay, thank you so much.
Thank you, sir.
Okay, ma'am. Thank you, ma'am.